Welcome to G Fun Facts Online, where we dive into the science that shapes our world. Today, we're going microscopic to witness a war that has been raging for billions of years, a silent, relentless battle fought between bacteria and the viruses that hunt them, known as bacteriophages. This is an evolutionary arms race on a scale we can barely imagine, and it has produced an arsenal of molecular weapons so sophisticated they're changing our own future. At the heart of this conflict is a system you might have heard of, CRISPR. But as we'll discover, CRISPR is just the opening chapter in a much grander story of bacterial defense. The famous CRISPR-Cas system is essentially a bacterial adaptive immune system. Think of it like a molecular scrapbook of past infections. When a virus or phage attacks a bacterium, the bacterium can capture a small piece of the virus's DNA. It then weaves this DNA snippet into its own genome, into a special region called the CRISPR array. This snippet acts as a memory. If that same type of virus ever attacks again, the bacterium transcribes this memory into a guide molecule, a small piece of RNA. This guide RNA then teams up with a protein, most famously Cas9, and acts like a molecular GPS, hunting for a matching sequence in the invading virus DNA. Once it finds the match, the Cas9 protein acts like a pair of molecular scissors, snipping the viral DNA and neutralizing the threat before it can take over the cell. But CRISPR is more than just a pair of scissors. Scientists have discovered a stunning variety of these systems. While some target viral DNA, others go after viral RNA. And some can even target both, and some have a much more dramatic strategy. A groundbreaking discovery revealed that certain CRISPR systems, instead of just cutting the virus, trigger a protocol of self-destruction. This is called abortive infection. For example, when a system called Cas12a2 detects a virus, it doesn't just cut the viral genes. It goes into a frenzy, becoming a shredder that indiscriminately destroys all DNA and RNA in the cell, including its own. The infected bacterium commits suicide, sacrificing itself to prevent the virus from replicating and spreading to its neighbors. It's a scorched earth tactic for the good of the colony. For a long time, scientists thought CRISPR and another defense called restriction modification systems were the main ways bacteria fought back. But recent discoveries have blown that idea out of the water. We're now finding a vast, previously unknown world of anti-phage mechanisms. These defenses fall into a few main categories. Some are about infection exclusion, basically putting up a